Okay, going back to facilitated transport or facilitated diffusion. In facilitated diffusion, you need a special carrier, a transmembrane integral protein, because the particles are moving under their own energy, but they're either too big or water soluble, not lipid soluble, so they need a special entrance. Good example, not the only, is glucose. Glucose going into a cell. Glucose is vital to be in a cell, so how does it get in? You have these transmembrane integral proteins called gluten receptors. We've classified 4 different kinds. Cells in the brain and other areas don't need insulin, but they still need gluten receptors, transmembrane integral proteins. The ones that need insulin to put the transporters in the membrane are skeletal muscle, heart muscle, and fat tissue. If insulin is not present, then the glucose that you've eaten cannot get into the cell and stays high in the blood sugar. High in the blood sugar. So you can read on that. Here's an example of a transmembrane integral protein allowing something that's called help transport or help diffusion. We now quickly go to active transport. Active transport is the particles do not have enough energy themselves. So they need extra energy from this substance called ATP. Now, I'm going to break active transport down into primary active transport and secondary. Primary and secondary. Okay, listen to me well. Every living cell in the human body, has something called the sodium-potassium pump. It has many of these pumps. Now, when you hear the word pump, that always means active transport. When you hear the word pump, that always means active transport. So every living cell in the human body has several copies, thousands of copies of this sodium-potassium pump along its membrane. Okay? That's what I'm going to call the primary pump. The way the pump works is, now let me come here. Sodium is an ion, so this is what you want to start writing down. Get a piece of paper. Sodium is an ion in higher concentration outside cells than inside. Sodium is an ion in higher concentration outside cells than inside. So you want to write this down, get a piece of paper, and draw your cell. Potassium is an ion in higher concentration inside cells than outside. Okay? What would happen is that over a period of time, sodium would leak into cells and potassium would leak out. Now, if that occurs, I'm going to come back here, we'll be talking about, if that occurs, then that will mess up something called our resting membrane potential. Now, here's something of importance in all cells, and particularly nerve and muscle. The resting membrane potential is a charge on the cell membrane. So I'm going to come here. I want you to read on this. On a cell surface, on a cell membrane, you have 2 gradients. Gradient means gradational. You have a concentration gradient and a charge gradient. The charge gradient is the charge on the cell membrane. The concentration gradient has to do with the ion concentrations. All right, so write this down. In a cell, the membrane on the outside has a net positive charge, and the membrane on the inside has a net negative charge. Outside, net positive, inside, net negative. So charges like to move. So the positive would like to move to the negative, and the negative would like to move to the positive. But the membrane holds a resistor, and it can't do that. So there are 2 gradients on a cell membrane. A concentration gradient and a charge gradient. A concentration gradient and a charge gradient. You'll have a video on this. Now, these gradients must be there. You need to have more positive on one side and more negative on the inside and so forth like that, and even the differential in ion concentrations to maintain the resting membrane potential. So if these items, charge and reached equilibrium, if they reached equilibrium, you would lose the resting membrane potential and death would occur. So let me say it again. It is vital to keep these ions at disequilibrium. It is vital to keep these ions at disequilibrium. But they eventually, through passive transport, would leak. The sodium would leak in and the potassium would leak out. Okay? So what has to happen? We must keep them at disequilibrium. So we must push the sodium from a low concentration to a high, which is against the gradient, and push the potassium the opposite way. So we must push from low to high. Thus active transport. The way active transport works is this. These sodium-potassium pumps push 3 sodiums out for 2 potassiums in. Let me say it again. It pushes 3 sodiums out for 2 potassiums in, which would kind of show you why there's more positive on the outside and less on the inside. But the sodium-potassium pump, every living cell has this, and it tries to keep these ions in disequilibrium for life. Now, I want you to read on this in the textbook and my PowerPoints. So the sodium, but every cell has it. So that's the primary pump. Now, because this is this way, and sodium's high on the outside, potassium on the inside. Now I want to draw your attention to sodium on the outside is positive. Okay? The inside the cell is negative. So sodium would like to come from its positive side to the negative. Okay, that's what sodium also is high outside and low inside. So sodium has a very hard driving force because its concentration gradient says go in, and the charge gradient also, since the inside is more negative. So sodium has quite a bit of energy. So much energy, listen to me, so much energy that something could piggyback on its exergonic excess energy. That comes the role of secondary transport. Secondary transport piggybacks generally on sodium. Okay? And I want you to read on it. Under secondary transport, you can have co-transport, also called symport or contraport, under secondary transport. Now, this is what I'm saying. If a particle wanted to come in to the cell, let's say the particle had low concentration on the outside, high on the inside. Now, particles have low on the outside, high on the inside, and they want to come in, they'd have to come in by active transport. But in certain cases, special transporters can occur where, since you know sodium is going to drive into the cell, it could piggyback something on its back. So when the sodium comes in, which it will, and has all that energy, it would carry the other particle with it. In that particular case, you wouldn't need extra energy. It would be a 2-for-1 deal. Since the particles are both going in the same direction, we can call that co-transport or symport. Now, on the other hand, suppose something was low on the inside and high on the outside, and you wanted to get that out. You'd have to pay for some more ATP. But you could create a special turnstile integral protein, so when sodium came in, it would flip the other substances out. When sodium came in, because it's exergonic, extra energy, it would flip the other substances out. Since the sodium would be going in one direction and the other material in another direction, we could call that contraport or antiport. Contraport or antiport, both meaning the same thing. So secondary transport is piggybacking on the efforts of primary transport, because every cell in the human body has sodium potassium pumps. I'd like for you to read on that in your textbook. All right. Now we go to large particle transport. Remember I told you the wall would have to be caved out. Okay, phagocytosis is called cell eating. You generally are bringing solid particles. Pinocytosis, cell drinking, and then you have a receptor-mediated endocytosis. Okay, here we go. When large particles are brought into a cell, it's called endocytosis. When you exit large and have to cave it out, it's called exo. So you don't use that for small particles. Use that for large particles, and you're clustering them up. Here's an example of phagocytosis. In phagocytosis, you engulf a particle. You create these false feet known as pseudopods. The substance comes in, and this closes around you and brings it into a vesicle or vacuum. Kind of read on that. It's bringing solid. Pinocytosis, you're making more liquid droplets. Come in, and you close behind it. Receptor-mediated endocytosis, you have special receptors, so you selectively bring in what you want, but you still have to cave it out. So all solid, all liquid, but special type here, because you have special receptors. So receptor enables the cell to acquire but it's specific. Embedded in the membrane are proteins with receptor sites. So it's very specific. I want you to read on that. Here would be it coming in, but you're hooking to these receptors and coming in. Okay. Exocytosis is when you push stuff out, like hormones, neurotransmitter substances. So read in the textbook. I'll try to also put a few videos in on that particular topic. Okay, here's exocytosis showing you. We won't get into the snares and everything, but I just want to show you exocytosis. That's to put large particles out. Endo, which was phagocytosis, pinocytosis, and receptor mediated was to bring them in. But we do have exocytosis also, as you can see here. Okay, we'll close this video here.